Hello everyone, I'm Professor Lee of Dr. Anti-Aging and Herbal Medicine TV. From today, we'll study the important things about each of the 12 types of cellular aging that we'll manage like disease. And for each topic, I'll summarize the latest aging treatment that you can do now that you are the most curious about in a prescription format. You can choose the method that seems right for you among the aging treatment introduced in this way and practice it. If you have any question, please let me know in the comment. First, cellular aging caused by DNA damage accumulation. Let's get started. In the nucleus of human cells, chromosomes are gathered, and what is densely condensed inside these chromosomes is DNA. If you enlarge the DNA part, it looks like a ladder with two strands twisted together. If you think of this entire ladder as a book, it has letters written in pairs of two letters. These letters are called bases. In humans, the entire document, made of about 3 billion of these base pairs, is DNA. Among the entire document, made of about 3 billion base pairs, DNA contains code letters that contain a recipe for making proteins necessary for life activities. These are genes. Of the entire 3 billion letters in DNA, only 20,000 to 25,000 code letters contains the genetic information for making proteins. Therefore, only 1 to 2% of the entire DNA is genes. Now, do you know the difference between DNA and genes? Among the DNA part, the gene part is called coding DNA because it is the code that creates over 100,000 proteins necessary for sustaining life. For example, insulin, male hormones, female hormones, thyroid hormones, and stress hormones are all proteins made with genetic recipe. Digestive enzymes such as amylase, lipolytic enzymes, and protein decomposing enzymes are also made with genetic information. And interferon necessary for immune response, collagen that maintains skin elasticity, and hemoglobin that transport oxygen are all proteins made with genetic information. What happens if the DNA part where the genes are located is damaged? Of course, proteins that perform the correct function will not be created. If there is an error in the recipe for cooking, the food will be tasteless. If the genes are severely damaged, then the food will be difficult to eat. The fundamental cause of aging, disease, and cancer is the accumulation of DNA damage, so it's very important to manage your DNA well. The remaining 98 to 99% of DNA does not contain information to create proteins, so it's called non-coding DNA. Non-coding DNA does not directly create proteins, but it has a regulatory function that promotes or suppresses gene activity. Telomeres, which protect the entire DNA, are also part of non-coding DNA. Since non-coding DNA accounts for a much larger portion than coding DNA, damage occurs more frequently, and when damaged, gene activity becomes unnecessarily strong or weak, which also results in harming health. You all know about genetic testing for paternity, right? If the DNA of a child and a parent matches 99.99%, they are diagnosed as a real family. However, the DNA used for paternity testing is not coding DNA, but non-coding DNA. The coding DNA part is the part that makes proteins essential for life, so there is not much difference between people. However, the non-coding DNA part is different for each person and has a slightly different unique patterns. So, if this part matches 99.99%, it's considered paternity. Even at this moment, each cell in our body experiences about 100,000 damages to its DNA every day. If you look at the entire body, trillions of damage occurs every day. This number is tremendously large, but fortunately, our bodies have mechanisms to repair and restore it. However, this process is not a perfectly precise operation, so it's natural that errors and damage occur. The most serious type of DNA damage is when the double strands are completely cut or tangled, and it is difficult to repair. Chemotherapy drugs are drugs that kill cancer cells by damaging their double strand, but they also kill normal cells, so they have side effects. They also occurs when exposed to radiation and very long period of harsh sunlight. If you are exposed to the sun for a long time, you really need to be very careful about UV protection. Single-strand damage is less fatal than double-strand damage, but it happens very often. It can occur due to errors in the DNA replication process and various environmental pollutants, heavy metals, smoking, and severe oxygen and radical stress are the main causes. That is why you should definitely wear a mask on days with a lot of fine dust. Aren't you curious about how damaged your DNA is right now? You can easily find out in 3 or 4 hours by taking a blood sample. For example, if you apply an electric force to the blood you draw, the DNA strand will move, and if there are any broken strands, they'll move and separate, forming a comet tail. This analysis method is called the comet analysis. This analysis method is one that I've used frequently for the past 20 years. When you test smokers with this method, they show twice as much damage compared to non-smokers. You can also find out if there is serious damage to the chromosome stem cells in the blood. If you look at the picture, you can see the fragment of the chromosomes in the white circle. They are fragments due to severe damage. In some cases, the damaged nucleus detaches and forms a small nucleus. This is called the micronucleus DNA damage analysis method. The most common type of DNA damage is damage to only the bases. Chemical modification of bases by active oxygens occurs very common every day, and exposure to ultraviolet rays and environmental pollutant is also a cause. Even if only part of the base is damaged like this, if it accumulates, it can distort genetic information and cause mutations, diseases, and aging. Damage to some of the DNA bases can be detected through urine tests, and many simple kits have been developed. If you drop an enzyme that binds to the damaged bases into your urine, you can measure the bound part. This is called DNA-based damage analysis. The small blue dots in the round cells in the portal are damaged DNA bases. If there are patients with high blood pressure who have more severe DNA damage, wouldn't it be great if they also take blood pressure medication and manage DNA damage? There is separate DNA in the mitochondria in the cells, and this is called mitochondrial DNA. Unlike the DNA in the nucleus, this DNA is not double helical, but round as you can see. I'll explain more in detail in the mitochondrial activation aging treatment section. Mitochondrial DNA is much more susceptible to damage and is less able to be repaired. I'll now explain the main causes of DNA damage. In order for cells to grow, DNA must be replicated during the division process. In order for the DNA double strand to be replicated, the two strands must first unravel and separate. When the two strands separate, a gap must be created so that DNA replication enzymes can squeeze in and create a new strand that appears orange using the original blue strand as a template. However, if the double strands separate like this, the DNA strand will become unstable, right? In addition, since the force of pulling and rotation is applied as they separate, the strand must break apart. However, since replication is necessary, it's an unstoppable damage. In addition, if the double strands separate and becomes a single strand, they are exposed to numerous damaging substances in the surrounding area, so they are easily damaged. However, replication must continue and finish, so if damage occurs during the process, replication stops for a while, and repairs are performed while replication continues, and then replication is completed. However, if there is any part that is not fully repaired during this process, the DNA that was replicated but was abnormal will remain. Another major internal cause of DNA damage is reactive oxygen. In particular, reactive oxygen is constantly generated when energy is produced in mitochondria, which are organelles within cells, and is actually the main cause of DNA damage. Radiation strikes DNA strands and breaks them, just like crashing a car on a highway, and repairing them is very difficult and carries a high risk of cancer. Clinical studies have shown that a single chest X-ray causes about 12 DNA single-strand damage per cell. CT scans expose the cells to much more radiation, causing thousands of DNA single-strand damage per cell and double-strand damage to cells in the blood. Using contrast agent during CT scans increases DNA damage even more. Therefore, taking antioxidants such as vitamin C before scanning significantly reduce DNA damage. Recently, low-dose CT scans have been introduced, which reduce DNA damage. We live surrounded by countless electromagnetic waves, in particular, high-frequency electromagnetic waves from cell phones, Wi-Fi, GPS, Bluetooth, and television broadcast, rather than low-frequency electromagnetic wave from home appliances and computers, can cause DNA damage when exposed for a long period of time. Therefore, research on DNA damage caused by cell phone use is also increasing. In the case of cell phones, there is no clear evidence of DNA damage at the general level of use. However, some studies have reported that if you sleep with your phone next to your head, the electromagnetic waves are exposed for a long time at close range, which can increase DNA damage. The sun's ultraviolet rays directly damage and change the structure of DNA. Clinical studies have shown that short-term exposure can repair it, but long-term exposure causes cell death and mutations. When skin is exposed to ultraviolet rays, the degree of damage increases with the exposure time. For example, if exposed to strong ultraviolet rays for 1 hour, the bases in DNA becomes abnormally attached and tangled. After 2 hours, the tangled structure double. Never forget how important it is to protect against ultraviolet rays when spending a long period of time outdoors. UVA, which has a long wavelength among the ultraviolet rays, penetrate deep into the skin, causing DNA damage, and is the main cause of skin aging. Among UV blocking creams, UVA blocking function is necessary to prevent skin aging. The blocking index is divided into 4 levels by PA, and for outdoor activities, it should be PA index 3, and for long-term exposure, it should be PA index 4. UVB has short wavelengths and penetrate only to the epidermis, causing redness, burns, and skin cancer. On the positive side, it increases vitamin D synthesis in the skin. The UVB blocking index of UV blocking cream is SPF, and for general outdoor activities, it should be used at 50 or higher. Processed food ingredient, waste combustion smoke, industrial processed smoke, and activating ingredient of anti-cancer drugs all damage DNA bases and cause mutations. Specific alkylating ingredients found in foods include nitrosamines contained in sausage, hams, and bacons, and heterocyclic amines generated from food cooked at high temperatures. And then there's aflatoxin, which is produced by moles on grains and nuts. Automobile exhaust fumes, fuel, coal, industrial dyes, residual pesticide, and aromatic compound produced during everyday high-temperature cooking are also common DNA-damaging substances that cause DNA mutations. In particular, cigarette smoke contains more than 7,000 chemicals, at least 70 of which are known carcinogens. These toxic substances directly attack DNA and cause mutations. A large-scale clinical study confirmed the accumulation of benzoyl, which causes DNA-based damage in blood sample of smokers, and the group that smoked for more than 10 years had DNA damage indicators that are 50% higher than those of non-smokers. Fine dust and other air pollutants, commonly encountered in urban areas, are also major causes of DNA damage. In particular, fine dust is a very small particle that can penetrate deep into cells, and its size is only about 1/30th of the thickness of the hair. In fact, when 2,000 adults living in areas with high fine dust exposure were tested, the average level of oxidized DNA bases was 35% higher. Alcohol consumption also has a major effect on DNA damage. Acetaldehyde, which is produced during the process of alcohol metabolism in the body, is toxic to DNA and can cause unrepaired mutations. Analysis of oral epithelial cells of people who drink heavily showed that the level of oxidized DNA bases increased by 60% compared to non-drinkers. People who drink heavily are more than twice as likely to develop esophageal and oral cancers, which is also related to DNA damage. Viral infections also play a role in DNA damage. For example, human papillomavirus invades cells, directly alters DNA, and causes mutations by interfering with the repair process. The level of double-strand breaks, a marker of DNA damage, is on average 70% higher in cervical cells from HPV-positive women than from negative women. This significantly increased the risk of developing cervical cancer. Other viruses, hepatitis B and C, have also been reported to promote DNA damage in liver cells, increasing the risk of liver cancer by 5-fold and 7-fold respectively. A variety of products used in everyday life are also reported to be associated with DNA damage. For example, butyrphenol and bisphenol, which are used in cosmetics, pharmaceuticals, and food and beverage processing, have been linked to DNA damage in sperm cells. Food preservatives and food additives are also known to cause DNA damage. There are so many things we must avoid if we want to keep our DNA safe. Thanks to the technology that can analyze the entire cellular DNA, we have learned that unrepaired DNA damage leads to mutations, and that throughout our lives, we accumulate about 10 to 50 mutations per cell per year. Of course, skin exposed to sunlight and lungs exposed to smoking accumulate mutations even more. DNA damage and the accumulation of mutations caused by it are among the most difficult to treat among the causes of aging, and clinical application is still in its early stages. Nevertheless, the first thing we need to do is to improve and strengthen DNA repair capabilities. Among these are organized methods that have been proven to prevent DNA damage in clinical studies, among things that can be done in daily life. In fact, if you consistently follow this lifestyle habit, DNA-based damage is reduced by an average of 20 to 30% compared to those who do not, so it's worth putting into practice. Make it a habit to wear a mask to prevent the fine dust, and don't forget to ventilate indoors frequently. Use sunscreen with the UV protection factor of 50 or higher and UVA protection level 3 or higher. Avoid long-term close exposure to your cell phones, especially avoid sleeping with it on by your bedside. Reduce exposure to heavy metals and pesticides by washing and choosing organic foods, and reduce processed food as much as possible. Quit smoking and drinking. Prevent cervical cancer, oral cancer, and pharyngeal cancer by getting vaccinated against human papillomavirus, and hepatitis B virus vaccination is very important for preventing liver cancer. Moderate aerobic and strength training activate enzymes related to DNA repair. Sufficient sleep also increases the speed of damaged DNA repair. The next part 2 lecture is about the problem of DNA damage repairability. In part 1 today, we mainly study DNA damage itself and prevention method. And in the next part 2, we'll study the problem of repairing what has already been damaged. And in the last part 3, we'll study about food, herbs, and pills that actually prevent DNA damage and enhance the ability to repair it. Thank you for your hard work for a long time, and I will see you in the next part 2.